Okay, grade 12. In the previous video, we looked at a lot in the previous video. Right? We looked at complementary base pairs. We looked at the structure of DNA. We looked at its location. We looked at its discovery. We looked at genes, chromosomes, histone proteins, and all of those types of things. Now we're going to look at very important topic for this syllabus is DNA replication. Alright, DNA replication is important because ultimately that is how we grow, dear grade 12s. DNA replication is important because that is how we grow, that is how we develop, that is basically how we live, right? If your body doesn't replicate, then you would have died a long time ago, right? Because old cells need to be replaced with new cells, etc., etc. But if they were to ask you to define it in exams, or if they gave it as a biological term, keyword is it is the duplication of a DNA molecule to form two identical copies or replicas. Where does this happen? In the interphase. Now the interphase is that phase of the cell cycle before a cell division. So if the cell would divide here, so the cell divides, the rest period is known as the interphase, and then it will have replication, and then the cell will divide again. And the reason is that the cell will divide into two, then the cell will replicate itself, and then it will go on and divide. So that is why replication must take place. And how does replication take place? So please go and look at any past paper where they ask you to describe DNA. I'm gonna come back to all these words, but I'm first gonna show you the diagrammatic representation. So what happens with DNA replication? What is happening? So the first part of DNA replication, right, is that the DNA double helix molecule, or the double helix DNA molecule, right? We know that DNA is double helix. We looked at its structure earlier on. It has a double helix structure like that, right? Now this double helix structure, this DNA, and it's important, people, you must say DNA double helix molecule. If you leave out double helix, it is wrong. Please, people, keep in your double helix. That must be a thing. When you're describing anything and DNA is involved, you say DNA double helix molecule. So this DNA double helix molecule is going to unwind, and it's going to form a sort of a ladder-like structure. When it unwinds, it forms a ladder-like structure, right? So it now forms a ladder, which we now call it's where it unwinds. Then thirdly, what happens? So the DNA double helix molecule unwinds. Then remember the weak hydrogen bonds? Now these weak hydrogen bonds will break, causing the DNA double helix molecule to unzip. Right? Remember the nitrogenous braces broke, so it's almost like a zip on your jacket, right? That is the jacket with the zip. When you pull the zip up, as you pull the zip down, right, it breaks into two like that, right? Something like that. So now the weak hydrogen bonds in between broke and it unzipped. So the DNA double helix molecule unwinds, weak hydrogen bonds in between will break, causing it to unzip, right? Now, this gives us, and I'm using a lot of extra words just so you can understand. This gives us two strands, two separate, what we call template strands, right? Two template strands. Two template strands, right? The reason I'm doing it in a different color is so that you can remember that these two are complements of each other. Right? Now remember, this is happening in the interface and it's also happening in the nucleus. Now in the nucleoplasm of the nucleus, there are free floating DNA nucleotides. Right? They are free floating DNA nucleotides. So those free floating, what do I mean free floating? It's just laying there in the nucleoplasm waiting to, bond with something. So those free floating nucleotides, here grade 12s, are now going to come to the template strand. Or let me rather continue in blue. Are now going to come to the template strand, and they are going to attach with their complements, whether it is G bonding with C, A bonding with T, T bonding with A, or C bonding with G. It does matter, but I'm not really going to indicate all of that as long as you know what I mean, and I'm sure you do, right? It's going to come, right, and it's now going to attach to its complement. And now this here, so now this one in red here, so this would have formed another strand, right? Now those two strands, the B strands, they are called complementary strands because they complement the template. So they are called complementary strands. And ultimately, the result now is that, so it's not a long process. And now ultimately we can say that the result of this process is that we now have two identical DNA molecules. And then obviously just remember to write at the end that process is controlled by enzymes. Don't forget that process is controlled by enzymes. Right? So let's just have a quick recap. The DNA double helix molecule unwinds. The weak hydrogen bonds in between will break, causing it to unzip, forming two separate template strands. Right? Free floating DNA nucleotides will complement the two separate DNA strands, forming complementary strands, resulting in the formation of two identical DNA molecules where the process is controlled by enzymes. Let's look at the words now that you understand it. So, the DNA double helix unwinds, weak hydrogen bonds between nitrogenous bases will break, forming two separate strands. Right? So, just remember it unwinds, it breaks, and it unzips. Right, forming two separate strands where both act as templates. Free floating DNA, they didn't put in DNA there, it's wrong, it must be DNA nucleotides in the nucleoplasm are used to build a complementary DNA strand onto each of the original DNA strands. You can also just write this part here. This results in two identical DNA molecules. Each molecule consists of one original strand and one new strand. Right? So, a lot of words, right? But ultimately, what is important here is what I explained to you. Is that the DNA double helix molecule, it unwinds. First fact. Right? The weak hydrogen bonds, between nitrogenous bases will break, and the DNA causing the DNA to unzip. Second mark. Right? It will form two strands it will form two template strands. Two separate template strands is fine. It will form two template strands, two separate template strands, or you can say now resulting in whichever way you want to put it, as long as you have the fact that it is now two separate template strands. Third mark. Free floating DNA nucleotides from the nucleoplasm complement the two template strands, forming complementary strands. Another mark. Resulting in two identical DNA molecules. You can add the other part where it says that two identical DNA molecules where each molecule consists of an OG. So can we see that each molecule, this would be the OG, the original, and then a new one. Right? The OG and the new. That is just what that is basically what they mean. The process controlled by enzymes. Finished. DNA double helix molecule unwinds, weak hydrogen bonds in between will break, causing it to unzip, forming two separate template strands. Free floating DNA nucleotides from a nucleoplasm complement each of the two template strands, forming two new complementary strands, resulting in the formation of two identical DNA molecules, where each molecule consists of an OG or an original molecule and a new molecule, where the process is controlled by enzymes, and you will get all 5 or 6. It can be 5 to 6 marks, normally 5 marks, depending on how they feel. Finished. If you need to replay, you can rewind and listen to my wording. 100% you will get 5 to 6 marks, I guarantee it to you. Right now, I just need to erase all of this. There we go. Alright, so then we have some significances or importances of DNA replication. Now, the significance is that it doubles the genetic material so that each cell receives the same amount of DNA during cell division, resulting in the formation of identical daughter cells during mitosis. You need to know this word for word, people. I'm not gonna so basically, what do we mean when we say it doubles genetic material? It means that we had that genetic material first, and now it made two, and then that is going to go on and on like that. Is what it means to double. And then, because it's replication, making identical copy, this has, let's just say, call it DNA X, which means that both of these will also have DNA X, which means that that will also have DNA X, that will also, and so forth and so forth and so forth. That is basically the simple explanation. But you need to know it word for word if they ask you in the exams. Please, this comes from exam guidelines. So this is when it comes from exam guidelines, or when it comes from memos, or not memos, or worksheets set by the department. Please look at how the questions are answered. Try to memorize it verbatim, but also understand, because what they are doing or what they are telling you is that, look, this is how I want to answer. That you're telling the marker, or the marker is telling you, or the department is telling you, I want it like this. If you give it to me like that, 4 marks in your bag. Right? Now we're just going to quickly look at DNA profiling. Right? It's not DNA fingerprinting. I said that in the last video. I made this notes a while ago, so it's still the old understanding, not DNA fingerprinting. It is just DNA profiling. So what is DNA profiling? DNA profiling has a barcode pattern, right? Not DNA fingerprinting. DNA profiling has a barcode pattern. It consists of non-coding DNA. Please go to page 1.5 of book 2 of the answer series to go and read up on non-coding DNA. I'm not going to go into it. You can go and read up on it, and the role of genes and all of that stuff. Please go and read up on that. Otherwise, these videos are going to be hours. But what is important to understand is that our DNA profile is unique to each person. I don't think I included an example here, but our DNA profile is unique to each person. And here we have the uses of DNA also from exam guidelines. Please, this is how they want you to answer it. Once I'm done, there's only going to be 3 videos. The next video, I'm going to stop this video here. And after I'm done explaining RNA, and then the next video will be protein synthesis, and then 2 to 3 videos after that will be exam questions, and I will go through those exam questions, analyze what I've heard are common mistakes from 2024. I look at exam guidelines, because please people, after every NAC, the department issues various documents stating silly errors. I've read through the documents, I've made notes of it, and I will go through it with you so that you don't make the same errors, guys. But anyway, so from exam guidelines, this is what it's used for: proof of paternity, tracing missing persons, to identify genetic disorders, establish family relations, match tissues for organ transplants, to identify dead persons or animals, to identify crime suspects in forensic investigations. It's a very common one. This is also a very common one. Those are actually, those four are quite common. Actually, those five are quite common ones, right? Because for the non-support, obviously, you need to do paternity testing, right? So from exam guidelines, please keep that in mind. There will be a video titled DNA profiling exam question, and then you're going to look at how all of it comes together. Now, finally, that was all of DNA. Now we're coming to RNA. Now, RNA is called, that's an error there. I think I was half asleep making this part of the note. It is supposed to be... wait. I think it's actually correct like that. If it's not correct, you know it's what if it's correct, if it's not correct. But I am not going to analyze whether it's... I think it is ribonucleic acid. I don't think it's ribose. I think I always say ribose. But anyway, I like to speak. The locations, right? It's found in the nucleus, which is mRNA, cytoplasm, tRNA, ribosomes, rRNA. You can remember that. It's easy to remember. I want to get here. This is where I want to be. There are 3 types of RNA. Firstly, you start with mRNA, which is known as messenger RNA. Now, what is important to note, this messenger RNA is formed in the nucleoplasm, and it carries a genetic code from DNA in the nucleus to the ribosome in cytoplasm. Right? So, from the DNA, which is housed in the nucleus, it will carry it out into the cytoplasm to the ribosome. That is what your mRNA function is. Right? That is the function of mRNA. That is the function of mRNA. Then, transfer RNA occurs in the cytoplasm. And also just note here that is something with mRNA, there's something called a codon, and we're going to look at it a bit later. It occurs in the cytoplasm. It has something called an anticodon, right? It picks up an amino acid, it takes it to a ribosome where several amino acids will bond via peptides, and it will, the process of protein synthesis will happen, and it will give you the required protein. Then rRNA, or ribosomal RNA, forms part of the ribosome, and it helps control protein synthesis. Quite simple to understand. I just want to see something here. Okay, so I just want to quickly look at this. This is the structure of an mRNA strand, or mRNA. This is the structure of a tRNA. I'm going to come back to that a little bit later. But what I want to get into, and I'm going to come back to this also a little bit later, but I want to quickly get into protein, or in the next video I'll get into protein synthesis. Otherwise this video is going to be too long. What I'll end off this video with is some differences between DNA and RNA. DNA is a double helix, first of all, right? RNA is not double helix, it is single, right? It is a single, single helix, or single stranded. You can also say double stranded and single stranded, right? DNA contains thymine as a nitrogenous base, where RNA contains uracil as a nitrogenous base. In DNA, adenine is always equal to thymine, and guanine is always equal to cytosine. In RNA, adenine is not equal to thymine, and guanine is not equal to cytosine. The ratio is unequal. And then another difference is that DNA has a deoxyribose sugar, RNA has a ribose Those are some differences between. Normally they only ask you up until 4. There are just a few things that I want to discuss quickly that I think I've left out. DNA, we know it's a double helix molecule. RNA is a single-stranded molecule, right? DNA, A bonds to T, G bonds to C. In that's actually very ugly drawing. In RNA, A bonds to U, G bonds to C. Note that in DNA it is thymine and adenine. In RNA it is uracil and adenine. Please, let's understand the difference. What are other important things that I think I left out? I think that is, yeah, I just said one is double helix, one is single stranded, one has T, one has U. And obviously the U and the C here are called pyrimidines. The A and the G are purines. TC is pyrimidines. AG is purines. That is important to note. And then in the next video, I'll stop there. In the next video, we'll do protein synthesis.